And we're going to go back to the notes that we began looking at the other day. So remember, we were talking about how eukaryotic cells make a protein. And we said there are 6 basic steps. The instructions are on the chromosomes in the nucleus. They're in the genes, to be specific. We make a copy of those instructions on a molecule called messenger RNA. We send that out to the ribosomes. The ribosomes basically read that messenger RNA and assemble the protein. We ship the protein around the cell in the endoplasmic reticulum. That's kind of our interstate highway system. And when we get to the edge of the endoplasmic reticulum that's pretty close to the Golgi apparatus, a little transport vesicle, a little lipid droplet, pinches off the endoplasmic reticulum and carries the protein to the Golgi apparatus. In the Golgi apparatus, the protein goes in the cystide, goes through several layers. It may go through all the layers. It may just go through some of them. But as it goes through each layer, it's modified by certain enzymes. Now, when we say modified, keep in mind what we're really talking about is the enzymes might remove an amino acid, or they might remove just a functional group on an amino acid, or they might add a functional group. They don't usually add amino acids. And then finally, the completed protein in its final form leaves the Golgi apparatus in a transport vesicle. If the protein is going out of the cell, we usually call the transport vesicle a secretory vesicle, because when a cell secretes something, it pushes it outside the cell. And then we said that 2 examples of vesicles are lysosomes, which are vesicles that come from the Golgi apparatus, and peroxisomes, which are vesicles that come from the endoplasmic reticulum. So now let's look at some of the parts of the cell that are not really directly involved in making proteins, but they are important for the cell to survive. And one of the things that we have to make proteins, or to do almost anything in a cell, is we have to have mitochondrion. Now, this is one of those words in science that has an unusual plural. Mitochondrion is singular. Mitochondria with an A is plural. A lot of times the mitochondrion is referred to as the powerhouse of the cell. I'm not sure that has a lot of meaning for most people. I think a better way to think of the mitochondrion is a furnace. And just like a furnace in your house burns fuel and uses the heat that's produced to warm your house in the winter, in this case, this furnace burns fuel. The fuel it burns is glucose. And it's not going to produce heat. It does make a little bit. That's why some animals like us are warm-blooded. But it's mostly going to be used to rebuild ATP, so that the cell can then use that ATP for anything it needs. So instead of warming the cell, we're going to mostly rebuild ATP in here, but it works like a furnace. We're going to burn glucose. We're going to combine it with oxygen. We're going to do it in a very slow, controlled manner, not like an open fire, but we are combining it with oxygen. Now, if we look at the mitochondrion, out around here, this white would be the cytoplasm of the cell. So remember, the cytoplasm is mostly water, but it's got a lot of things mixed in with it, so it's more like a thick milkshake than thin, runny water. The mitochondrion is separated from the cytoplasm out here by a double membrane. So there's this dark orange. This is the outer membrane. And notice that a bunch of the outer membrane and inner membrane have been sliced away so that we can see the inside. So this black line running around the outside, or this orange, represent the outer membrane. Notice the outer membrane is nice and smooth, has a very even surface. The inner membrane, if we look, has all these kind of ridges or all these fingers that stick into the middle. So the inner membrane has these little ridges, and these are called cristae. So those are those little parts that kind of stick into the center. And the center of the mitochondrion is filled with a fluid. So remember, we saw something similar with the nucleus. We had two membranes around the nucleus. We called them the nuclear envelope. And inside was the nucleoplasm. The fluid in here is called the matrix. And again, the matrix is kind of similar to cytoplasm or similar to nucleoplasm in the sense that there are a lot of water molecules there, but the matrix is specialized. So it also has chemicals related to the metabolic reactions that happen in the mitochondrion, in the matrix. And similarly, the nucleoplasm has a lot of chemicals that are involved in the chemical reactions that happen in the nucleus. So they're similar to the cytoplasm, they're not exactly the same. Now, if we look at the mitochondrion is unusual. If we look at eukaryotic cells, one of the things we see is that the chromosomes are inside the nucleus. They're contained by that nuclear envelope. But if we look in the matrix of a mitochondrion, we actually see a chromosome. But it's not the type of chromosome you might expect to see. First of all, there's only one chromosome. Remember, we've said eukaryotic cells usually have an even number. There's only one chromosome, and it's a circular molecule. It's not linear like we see in eukaryotic cells. And when we analyze the molecule, it's also naked DNA. There's no protein associated with the DNA. Well, one chromosome, circular, naked DNA, this is a prokaryotic chromosome. Those are all characteristics of prokaryotic chromosomes that we talked about. So it's got a chromosome in here, but it's a prokaryotic chromosome, a chromosome like you would find in a bacteria cell. And not only that, but if we look at the matrix, there are ribosomes in the matrix. But they're not like the ribosomes that are out attached to the rough endoplasmic reticulum or the cytoskeleton in your cell. The ribosomes are a different size. They're not the size of eukaryotic ribosomes. They're the size of prokaryotic ribosomes. And when we look at mitochondria, they actually reproduce separately from the rest of the cell. They can grow and then divide in half. So they can split into two mitochondria. And when they grow and divide, they don't grow and divide like eukaryotic cells do. They grow and divide like prokaryotic cells do. And there are some antibiotics that affect mitochondria that affect bacteria cells, prokaryotic cells. So as evidence started to accumulate, a hypothesis was proposed, and it's now been tested in a lot of experiments. So we call it a theory. Now remember what we said about theories in science in the first chapter. When we call something a theory, it's been tested in many experiments. We haven't been able to reject that hypothesis, so we're pretty sure it's right. This explanation for why we have a prokaryotic chromosome and prokaryotic ribosomes, and the cell divides like prokaryotic cells do, and several other characteristics, this is something called the endosymbiotic theory. Now, symbiosis occurs when organisms live together. So there are examples of symbiosis, for instance, when insects pollinate plants. That's a type of symbiosis. Endo means inside. So the endosymbiotic theory says that mitochondria are prokaryotic cells We think we know the group of bacteria they're related to by doing DNA analysis. The endosymbiotic theory says these are prokaryotic cells that have evolved to live inside eukaryotic cells. Now you might think, how could this happen? But we know that eukaryotic cells often eat prokaryotic cells, but sometimes they kind of swallow a prokaryotic cell, if you want to think of it that way, but they don't actually digest it. So it is possible that a prokaryotic cell could get into one of your cells and survive. In fact, that's what happens with certain types of diseases. So the mitochondria are crucial because the mitochondria are going to make most of the ATP in a cell. And in fact, there are a lot of diseases that occur in people if you have some kind of genetic defect in your mitochondria, if they don't make enough ATP. Depending on how bad it is, some of these diseases are actually fatal in a fairly short period of time. Now, remember something that we said. When we look at ribosomes, some of the ribosomes are attached to the endoplasmic reticulum. Some of them look like they're just floating in the cytoplasm, but they're not really. They're attached to something called the cytoskeleton. So you could think of the cytoskeleton as being kind of like the skeleton in your body. If we look at the skeleton in your body, we have bones, we have ligaments, we have tendons, and different things are attached to these bones and ligaments and tendons. In the cytoskeleton inside a cell, we have three different types of proteins. We have some microfilaments that are made of a protein called actin. And micro means small. A filament is something that's like a thread. Now, actin is actually kind of a circular molecule. It kind of has a ball shape. But if we look at a microfilament, notice that a microfilament is two strands of actin twisted around each other, and each strand is made up of a chain of these little actin balls. Think of it like a necklace of pearls or something like that. We have all these little circles strung together. We have two of these, and then we twist them around each other. That's how the cell makes a microfilament. We also have things called intermediate filaments. And when you think about intermediate means in between or in the middle. And these can be made of a protein called keratin, which is also found in your hair and your skin and your nails. A good example of how the same protein is used in different ways. But there are some different proteins that are part of the intermediate filaments too. Depends on what type of cell you're looking at. So we're going to mention that the intermediate filaments are made of keratin because that's probably most common, but keep in mind, there could be other proteins involved. In this case, keratin is a long, stringy molecule. So what we have is several molecules of keratin, and they're twisted around each other. Now, this is very similar to if you've ever looked at a rope. Most ropes are not made of a single strand of material. They're actually made of several strands twisted around each other. So are threads, though on a much, much smaller scale. So when you look at an intermediate filament with a good enough microscope, you can see these twists. And each strand is a molecule of keratin or other types of molecules. Finally, there's a third possibility, which is microtubules. Now notice, microfilaments are the smallest, microtubules are the biggest, intermediate we said was in the middle. In terms of their size, intermediate filaments are kind of middle. They're not as big as microtubules, they're not as small as microfilaments. So when we look at a microtubule, microtubules are made of a protein called tubulin. And tubulin is like actin in the sense that one tubulin molecule is actually circular. Now, in a microtubule, what we have is this chain of tubulin molecules. Again, it's kind of like a necklace of beads or pearls. This chain is twisted into a kind of looks like a can because it's actually got fluid on the inside. Um, so this structure has a little hollow area on the inside that can be filled with fluid or something else. So if we look at a cell, the purple here represents the plasma membrane. So this is the edge of our cell. So the external environment, whatever is outside the cell, is out here. The blue in here is the cytoplasm. All these little lines running throughout the cytoplasm, these are parts of the cytoskeleton. So they're either microfilaments or intermediate filaments or microtubules. And they're trying to show something that I mentioned briefly. We look down here in kind of the lower left, this pinkish with the little dots on it. This represents some rough endoplasmic reticulum. Those little dots are ribosomes. So the rough endoplasmic reticulum is actually kind of anchored to the cytoskeleton here, and it's anchored at other locations as well. Up here, we have a little part of a mitochondrion, and the mitochondrion is anchored to this section of the cytoskeleton. So when we look at the little organelles in the cell, they're not really just floating around haphazardly. Really, when we look at these, what we find is that they're anchored to different parts of the cytoskeleton. So we want to keep that in mind. Some people think that the cytoskeleton is in a sense the most important part of the cell, even more important than the nucleus, in the sense that this is what gives the cell some structure. Now, notice that these molecules that are part of the cytoskeleton, it's very easy for the cell to add on a few more tubulin molecules. I said, think about this like a necklace. You can stick a few more beads on the end. The cell can also take off a few more molecules by snapping a couple of tubulin molecules off the end. Can do the same thing with actin. So this means that the cytoskeleton can also help the cell move or change shape because certain parts of it can get longer or get shorter. And when they do that, the cell's going to change shape. It may grow, it may shrink. So we want to keep that in mind. Now, this protein that's found in microtubules, this tubulin, that turns out to be found in several different locations. One of the places that we find tubulin in cells is in a location called the centrosome. And the centrosome is made of two centrioles, and the centrioles are oriented at right angles to each other. So if this one is going left-right, this one is going front-back. When we look at a centriole, all these little orange lines in here, all those little orange lines represent microtubules. So each one of those orange lines is one of these coils with the little spherical molecules on it. Now, in a centriole, there are 9 of these around the center, and there are actually 9 pairs. And the reason we say 9 pairs is they really do occur in pairs. We don't see just 18 of them. So there are 9 pairs, and then there's 1 pair in the middle. So because of this, we say that this structure is something called a 9+2 arrangement. And we find this in not just the centrioles, but some parts of the cell called undulopodia. And undulopodia include 2 things, cilia and flagella. So if we take an undulopodia and we kind of slice through it, and we look at these molecules, this is what we see. So here's 9 pairs around the outside. Notice that one of the molecules has something called dynin attached to it. Dynin is kind of like a motor protein. It's going to move this. And in the middle, we have a single pair. So this is 9 plus 2. 9 pairs plus 2 in the middle. I don't know why it's not 9 plus 1, because there's 1 pair in the middle, but it isn't. It's 9 plus 2. Now, cilia and flagella can help a cell to move. When we discussed bacteria a few days ago, we talked about the flagellum on a bacteria cell. And we said on a bacteria cell, the flagellum turns around, kind of like a propeller on a boat motor or a plane, before there were jets anyway. And when we look at the flagella and the cilia that we see in eukaryotic cells, they work differently. They don't twirl around like a propeller. They kind of wave back and forth. If you've ever seen a fish tail when the fish swims, they kind of move back and forth like that. Now, cilia and flagella both have this 9 plus 2 structure. So how do you tell them apart? Well, cells that have flagella usually have 1 or sometimes 2 or 3. They have a couple at the most. Cells that have cilia often have hundreds or thousands. So they have a lot of them. Also, flagella are much longer. Cilia are very short. Here's a cell that's covered with cilia. This is all one cell. It's magnified. So this dark area in the center is actually the nucleus. But if you kind of focus on the edges, you can see these little green hairs sticking up. These are the cilia. This is a microscopic organism called paramecium. I know some of you said you'd had other biology classes before, so you might have looked at paramecium in the microscope. It's a pretty common organism to study. Now, when we look at these, remember I said that they're going to wave back and forth like the tail on a fish. And the tail on a fish, when it waves back and forth, this is what makes the fish swim. And in the case of cells, when the cilia or the flagella on the cells wave back and forth, this makes the cells move. So some cells can kind of swim through their environment, like paramecium can swim through its environment. It has a weird body shape, so it doesn't move in a straight line. It kind of goes in a corkscrew shape. So when we look at cilia and flagella, we don't see them in a lot of cells in humans. We only see flagella, or maybe I should say a flagellum, in one type of cell in humans. Does anybody know what this cell is? It is a cell that swims to where it needs to go. The only human cell that has a flagellum that swims to where it needs to go is a sperm cell. We find cilia in 2 different locations in the human body, but the cells that have cilia on them are kind of anchored in place. They can't move. So when the cilia wave back and forth, when they flip around, the cell doesn't move. Instead, they create a current. You know, if you've ever been in a boat and the boat is tied up to a dock, if it's a boat like a canoe that you're going to paddle, if the canoe is out loose on the lake and you paddle it, the canoe moves. If the canoe is tied up to the dock, the canoe doesn't move, but you basically push water past the canoe. And so we have 2 areas in the human body where we see cilia. Cilia line the upper part of your respiratory tract. And so when stuff gets down into a tube called the trachea that eventually leads to your lungs, you have cilia, and the cilia are always flipping stuff upward. So they're pushing junk out of your lungs. When they push it up into the back of your throat, you swallow it, it goes down your esophagus to your stomach. And those strong stomach acids that we talked about the other day dissolve this stuff that was trapped going down to your lungs. This is actually one of the main problems that people who smoke have. Smoke produces a byproduct called tar, and the tar clogs up the cilia in the lungs, so they don't push as much stuff out of the lungs. So a lot of studies have shown that people who smoke are more likely to get pneumonia, bronchitis, all sorts of lung infections because of the fact that their cilia don't clear the lungs as well as they could. The other place where you find cilia in the human body is in some place called the fallopian tubes. And the fallopian tubes are two tubes, one on the left side, one on the right side. They're only found in women. And the fallopian tubes connect the ovary to the uterus. Now, the ovary is where the egg is produced. And when the egg is released during ovulation, the fallopian tubes are hollow tubes. And when the cilia wave back and forth, they create a current. And the current gradually pushes the egg cell down towards the uterus. And the uterus is where that egg cell is going to attach and form a placenta if the egg is fertilized. Now, notice when I say that the cilia wave back and forth and create a current, keep in mind it's not a very strong current. The fallopian tubes are maybe about an inch long, and it takes the egg 7 days, maybe 10 days to get from the ovary down to the uterus. So the current they create is only moving the egg about 1/7 of an inch every day, so not very far. But they do create a current. So in big animals, in multicellular animals, where many cells make up their body, you might have some cells with cilia or flagella that are anchored in place. And when the cilia or flagella wave back and forth, they create a current. In some small animals, the cilia or flagella is how the whole animal moves, or in individual cells like sperm cells. All sperm cells, almost, we can't say anything for everything with living organisms, but almost all sperm cells swim to the egg cell using flagella. And there are actually some types of organisms that produce egg cells with flagella too. So the egg cells swim, not just the sperm cells. So where do we find tubulin, that protein that we talked about before? Well, we find it in the microtubules that are part of the cytoskeleton. We find it in the centrosome, or the 2 centrioles, if you'd prefer. And we find it in undulopodia, cilia, and flagella. So again, a good example of how in nature the same protein may be used over and over again for different functions. It's not necessarily always used in the same way. So we want to keep that in mind. Now, when we look at eukaryotic cells, we think of plants as being very different than animals, and they are in some ways. But at the same time, they're both eukaryotic cells. They have a lot of similarities. So before we leave the topic of eukaryotic cells, I want to think about what exactly are the differences and the similarities between plant cells and animal cells. And notice that we're talking about kind of a generic plant cell and a generic animal cell. There are over 240 different types of cells in the human body. And they aren't all exactly the same. If they were all exactly the same, there would only be one type of cell. And there are different types of cells in a plant body. So let's look first of all at what animal cells have that plant cells don't. And they really have two main things that plant cells don't. They have those centrosomes that we just talked about. And those centrosomes are going to help the cell divide when it divides by mitosis or meiosis, two terms that you might have heard if you had biology before. And animal cells have lysosomes. Plant cells don't have either of these. Now, plant cells do have a different type of structure that organizes their microtubules, because that's what the centrosomes do. But they're not called centrosomes. They look a little bit different. I also think maybe there's some territoriality involved. Since they're a little bit different than what we see in animal cells, botanists didn't want to call them the same thing that they had always been called in animal cells. So they gave them a new name so they could say they were different. And plant cells don't have lysosomes. They do have a certain type of vacuole that works just like lysosomes work. So some people even call them lysosomes, but again, if we look at their chemical structure, their molecular structure, they're a little bit different than what we see in lysosomes. They do both contain those hydrolytic enzymes, enzymes that take big molecules like proteins and break them down to smaller molecules such as amino acids. So they are very similar. They're not exactly the same. So again, we usually give them a different name. On the other hand, plant cells have three things that we don't see in animal cells. One of the things they have is something called chloroplasts. Chloro means green. And there are several different types of membranes called plastids. Now, plastids are membrane-bound organelles. A chloroplast is a membrane-bound organelle that's separated from the cytoplasm by a double membrane. So notice this is three organelles that we've talked about now that are separated from the cytoplasm by a double membrane. The nucleus, that nuclear envelope, was a double membrane. The mitochondrion a few moments ago, and now the chloroplast. Chloroplasts have a chemical called chlorophyll in them. And chlorophyll, we just said chloro means green, phyll means leaf. This is literally what makes leaves green. And this is really, for most plants, the only part of the plant that's green. There are a few plants that are exceptions. Now, what do chloroplasts do? Chloroplasts convert light energy into chemical energy of chemical bonds. And they do this during a process called photosynthesis. Every living thing that carries out photosynthesis has chlorophyll in it. Some of them have other chemicals that help the chlorophyll, but they all have chlorophyll. And so chlorophyll is crucial even for humans. We may not have chlorophyll in our cells, but all the food that we get comes from plants. Even if you're the opposite of a vegetarian and you only eat meat, if you're eating a hamburger, that cow ate grass. So if we trace back your food far enough, it came from plants. Now notice, similar to mitochondria, chloroplasts have their own chromosomes, their own ribosomes, and so we think that chloroplasts evolved from certain prokaryotic cells, certain bacteria, just like mitochondria did. And this is what the endosymbiotic theory says. So this is one of the things that we find in plants that we don't find in animals. Now notice, chloroplasts are a specific example of something called a plastid, but there are different types of plastids, and some plastids are found in animals, but not chloroplasts. Plant cells have a cell wall made of cellulose. Now, if we look at our plant cell, the cell wall is this greenish on the outside. The yellowish that's right inside that is the plasma membrane or the cell membrane. So the cell wall is outside the plasma membrane. And in fact, the plasma membrane is accepted as the last part of the cell that's really alive. The cell wall isn't alive, even though it's part of the cell. It's basically material that's pushed outside the plasma membrane. It doesn't carry out any metabolism. It doesn't do any of the things that we associate with living organisms. And finally, plant cells have a large central vacuole. Now notice there are lots of central vacuoles, lots of different types of vacuoles, kind of like lots of different types of plastids. And some of the different vacuoles are found in animals, but not a central vacuole. So the central vacuole is the big kind of swimming pool looking thing here in the middle. And when plant cells are small, they often start out with a couple of small, what are called water vacuoles. And you might guess by hearing the name, water vacuoles mostly have water in them. And the water vacuoles coalesce together, kind of like droplets of water, running down a windshield that join. So these water vacuoles join together, and when they join together, they make the central vacuole. And the central vacuole is very large. You can kind of see from this picture. The other parts of the cell are kind of pushed out right around the edge, close to the plasma membrane, because that central vacuole is in the middle. It occupies so much space. Now, since it was made by combining some water molecules, there's a lot of water in here, but it does store chemicals besides water. A lot of times there are pigments, there are colors in here. So when you look at an apple peel, whether it's a yellow peel or red or green, it looks like the color is everywhere, but really the color is concentrated in the central vacuole. Similarly, a lot of times there are natural insecticides in this central vacuole. Since this central vacuole is big, this means if an insect bites into this central vacuole in a plant cell that's part of a leaf or stem, it's going to get a bite of this insecticide. So this is one way to try to fight off things that are eating the plant. There are a couple of plants like milkweed that do produce poisons that are toxic to vertebrates, the group of animals that we're in. So milkweeds make a chemical called cardiac glycosides. And cardiac glycosides essentially could give you a heart attack. So if you're tempted, don't ever eat monarch butterflies or caterpillars. So plant cells have a cell wall, they've got a central vacuole, and they've got chloroplasts. So they've got three main things that we don't find in animals. Animals have two main things that we don't find in plants. When we look at animals, animals have lysosomes and centrosomes. Now, we have to keep in mind for these two things, although plant cells don't have exactly the same thing as centrosomes, they don't have exactly the same thing as lysosomes, they do have little organelles that do the same job. Animals, on the other hand, don't have any organelles that do what chloroplasts do. If you had something that was like a chloroplast, you could go sit outside on a nice sunny summer day. Not today, but you could go sit outside on a sunny summer day and you could make your own food. Plant cells have a cell wall made of cellulose. Animal cells don't have a cell wall at all. and plant cells have this large central vacuole made from smaller water vacuoles. Animal cells do have a couple of types of vacuoles, but they don't have anything that's the equivalent of this large central vacuole that takes up most of the space in the cell. Now, the other parts of the cell are basically the same. Here in purple, we have our nucleus. The red in the middle is the nucleolus. These little layers out here are the rough endoplasmic reticulum. Here we have some smooth endoplasmic reticulum. Here we have the Golgi apparatus. Now notice this diagram says there's a lysosome here. Remember what I said, some people call it the same thing. We have mitochondria. One way to tell a mitochondria from a chloroplast is mitochondria usually have little lines in the middle. It kind of reminds me of like a tennis shoe footprint, if you will. So really, except for a few organelles, chloroplasts and mitochondria have the same types of structures. So even though we think of plants as being very different than animals, remember that was kind of how we started out this part of our discussion, plant cells and animal cells are actually very similar. There are a lot more similarities than there are differences. The differences are important. You know, plants can't move. Some people think that's because of their stiff, thick cell walls. Animal cells can, because all we have is a thin, flexible plasma membrane around our cells. Are there any questions about plant cells and animal cells, or anything else that we've talked about with cells so far? Okay, we're going to stop sharing that, and we're going to start on something that isn't cells. So you might say, why are we going to talk about things that aren't cells? Because we've said that all living things are made of cells, and this is biology, the study of life. But we need to talk about a few things that aren't really made of cells, but they can interact with cells. And these are three types of things called viruses and viroids. Remember, we said oid means like, so viroids are kind of like viruses, and prions. Now, viruses, viroids, and prions are not considered to be alive. And the reason they're not considered to be alive is they only have two of the characteristics of life. These organisms can reproduce. However, they can only reproduce when they're in a living cell. And when viruses reproduce, they can mutate and evolve. But notice, none of these organisms have metabolism. None of them use energy. Some of them don't even have DNA. So they don't have all the characteristics that we think of living organisms as having. So let's look first at a virus, and let's think about a virus that's been in the news a lot in the last year and a half or 2 years. Let's look at SARS-CoV-2. SARS-CoV-2 is actually the name of this virus. COVID-19 is the disease it causes. Well, when we look at viruses, there are only two things that all viruses have. So if we look at this virus, you can see these little rectangles that are different colors that form this kind of curlicue towards the middle. And these little rectangles for this virus are actually RNA nucleotides. Now, some viruses have DNA, but SARS-CoV-2 has RNA as its genetic material. Now, notice this molecule of RNA is not called a chromosome. So we don't say that viruses have chromosomes. Some viruses have RNA, some have DNA, and whether it's RNA or DNA, it does the same thing. The nucleic acid just like we talked about with prokaryotic cells, just like we also talked about with eukaryotic cells. This nucleic acid can be divided into sections that we call genes. And each gene is the instructions for the primary structure of a protein, the order of the amino acids in a protein. Now, a virus normally has these instructions. So it has some nucleic acid, either RNA or DNA. Depends on the virus. Some viruses have one, some have the other. There are no viruses that we've discovered so far that have both. You know, eukaryotic cells, even prokaryotic cells, have DNA and RNA. So we want to keep that in mind. So here we've got one or the other. And then you can see this little blue line that's kind of curling around that the nucleic acid is attached to. This is something called the capsid. And the capsid basically surrounds the nucleic acid, whether it's DNA or RNA, and protects it. Now, when the nucleic acid is attached to the protein that makes up the capsid like this, then the protein is sometimes called nucleoprotein or just N protein for short. This is the only two things that we have to have in a virus. When we look at viruses, viruses have nucleic acid. The nucleic acid is the instructions to make proteins. We have a protein capsid, and these are the proteins that the virus has the instructions for. So notice this is a little, that we can't call it living, but this is a little factory that all it does is reproduce. It has nucleic acid that has the instructions to make proteins. When it makes the proteins, that protects the nucleic acid. So there's no point to this other than reproducing. Now, some viruses have a layer out here that they pick up from the cells that they infect, their host cells. And this is the virus envelope, or it's sometimes called the virus membrane, but it's not exactly the same as the plasma membrane on a cell. This membrane is something that the virus steals from the cell that it infects when it breaks out. So some viruses take a part of their host cell's plasma membrane with them. Some viruses just punch a hole in the plasma membrane of the host cell. And when they punch a hole in it, they rupture the cell, and usually that kills the cell. Now, stuck in this viral envelope, if we have one, because we don't have to have one, but stuck in here are proteins for the virus. So this big purple structure represents that spike protein that you see whenever you see the pictures of SARS-CoV-2. But there are several other smaller proteins in the area. And there are sometimes some enzymes found in here. Remember that enzymes control metabolic reactions. And since a virus doesn't have exactly the same function as a cell does, when a virus gets into a cell, a virus is going to use the cell to reproduce. But sometimes the cell doesn't have everything the virus needs. So if the virus needs an enzyme that a cell doesn't have, then the instructions for making the enzyme will be in the nucleic acid, because remember, an enzyme is just a type of protein. So the instructions for making the enzyme are in here. So before the virus leaves its host cell, it will make some of these enzymes. So then they'll be available when it infects another cell. So let's think about why a virus can't survive on its own. Because a virus can only reproduce when it gets inside another cell. Now, we might think of viral illnesses like COVID-19 as only affecting people, but other animals do get virus illnesses. There are some viruses that infect plants. There are even viruses that infect bacteria. So, when we study living things, if we study them long enough, we almost always find out that there's some kind of virus that infects them. So let's think about how a virus actually infects a cell, and let's look at the steps. And there's 6 main steps. Some of the viral proteins. So notice that would be the proteins in the outer layer, or if we had a virus that didn't have this outer layer, because remember, some viruses end with this blue line. If the blue line is the outer part, remember that this blue line is protein. So we're either talking about proteins out here, or we're talking about proteins in here. So the viral proteins bind to receptors on the outer surface of the host cell's plasma membrane. Different host cells have different receptors. So viruses can only infect host cells that have receptors its proteins can attach to. Well, what decides whether its proteins can attach to the receptors? Shape. Remember, it's always 3-dimensional molecular shape for molecules. So for instance, when we look at SARS-CoV-2, the spike protein has to attach to a protein called the ACE2 receptor. Now, that means it can't infect cells that don't have an ACE2 receptor. Unfortunately for us, lots of different types of cells in the human body have an ACE2 receptor. Now, once it attaches, the virus enters the host cell. Depending on the type of virus and the type of cell, sometimes the whole virus enters the host cell. Sometimes the virus just pushes its nucleic acid through the plasma, and the protein capsid, the membrane, if there is one, or envelope, stays on the outside. If the entire virus goes in, then the next thing that has to happen is we have to take off that viral capsid. So remember that capsid is the blue curlicue of proteins. We have to take that off so that we get our nucleic acid all by itself. Now, at this point, the virus is going to use the host cell to reproduce. So when we make a copy of nucleic acid, we say that we're replicating the nucleic acid. So what do we need to replicate nucleic acid? Well, think about making more DNA or RNA. The virus could be either. We need nucleotides because we're going to make a molecule of DNA or RNA. We need enzymes to control the chemical reactions where we're going to put these together. If we're going to put them together, we need energy. We need ATP in the cell. Viruses don't have any nucleotides except for the ones that are already part of their original DNA or RNA. They don't have most of the enzymes they need. They don't have any ATP. So all this stuff, the enzymes for the chemical reactions, the energy for the chemical reactions, the nucleotides, which are going to be the reactants, all of those things they get from their host cell. Now, we've talked about proteins a little bit, but think about what you need to, make proteins. You still need enzymes. You still need ATP. You still need amino acids. Ribosomes don't have any of these things. They might have 1 or 2 of their enzymes, but they don't have all of them. So where do they get all this material? from their host cell. So we've said that cells are organic factories that make proteins. So when a virus infects a cell, it's sometimes been described as hijacking the cell. The cell quits making its own proteins and starts making virus proteins. The cell quits making messenger RNA and DNA for itself and starts making virus messenger RNA or DNA. Now, sometimes when the DNA or RNA is being replicated, there's a mistake. You know, you could think of replication as like I hand you a set of notes and I say, I'd like you to copy these over on another sheet of paper. Well, you know, it's possible that you could make a mistake. When a mistake is made, this is something called a mutation. Now, in a mutation, the nucleic acid isn't replicated accurately. So remember, we've said that we've got those 4 bases, A, C, G, and T, or A, C, G, and U, depending on whether we have DNA or RNA. So replication isn't accurate. But remember, the order of the nucleotides tells the cell the primary structure of its proteins. It tells the structure, the order of the amino acids in the proteins. So if you change the DNA or RNA, whatever the virus has, not only do you change that, but that also changes the proteins. Well, when you change the proteins, notice that you're changing maybe this spike protein or maybe one of these other proteins, these little green blobs in here. Now, why does this matter? Well, remember what we just said. This spike protein, this is what attaches to a protein on the outside of a cell. This is how it, in a sense, picks which cell it can infect by where its proteins fit in. So if we change the amino acids, we might change where this virus can infect. But there's another problem. When we look at your immune system, the way that your immune system fights a virus or a bacteria, say we're talking about the SARS-CoV-2 virus, the way your immune system learns to fight this is by recognizing the shape of this protein. And once your immune system learns to recognize the shape of this protein, then you have cells whose job is really just to travel around your body and look for this protein and attack it. So what happens if we change the shape? Well, if we change the shape of the protein enough, the cells in your immune system don't recognize this protein when they encounter it. And so this is what happens, for instance, if you know about the flu vaccine. If you get the flu vaccine this winter, it probably won't be good next winter. Why won't it be good? Well, because the virus that causes the flu turns out to mutate very quickly. So over a whole year's time, when the virus comes around next year, some of the proteins on the outside are going to be different than they were last year. And so your immune system is going to be going around looking for last year's flu virus, but it's not going to find it. So when they make the virus, what do they try to do? Well, the flu virus usually starts in the Far East, in Hong Kong or Singapore or Japan. So scientists usually look at what version of the flu virus is most common, and then that's the type that they use to build the vaccine. But every once in a while, we use a version of the flu virus to build the vaccine, and then by the time it gets to the United States from the Far East, there's a different version of the flu virus that's dominant. So sometimes the vaccine doesn't do very much good. So once we make all our proteins, all our nucleic acid, then the protein and nucleic acid are assembled into a complete virus. And these new viruses are released. Sometimes, as I said, they kind of pinch off the cell, taking part of the plasma membrane with them. This is their envelope. Sometimes they just burst through the plasma membrane and punch a hole in it. And depends on which type of virus it is. Each virus has a characteristic, so a virus doesn't really change this. So when we look at the immune system, how do we fight viruses or bacteria or certain fungal diseases like ringworm or parasitic worms? Well, we have some cells called macrophages and neutrophils that phagocytize viruses and bacteria. And phagocytize is basically the biological term. They basically eat viruses and bacteria. And once they eat them, then they combine them with a lysosome. So remember, lysosomes are those membrane-bound organelles in the cell. They contain hydrolytic enzymes, which are sometimes also called digestive enzymes. And these enzymes take proteins, break them down to amino acids, take nucleic acids like DNA or RNA, break them down to nucleotides, take polysaccharides, break them down to monosaccharides. So they take large, complex molecules, break them down to smaller, simple molecules. And when they do this, when they break down the proteins in a virus to just amino acids, the virus doesn't function normally. It doesn't work anymore. Now, we also have some cells called B cells. And B cells get their name because they mature in your bone marrow. And these cells make something called antibodies. And antibodies, of course, are proteins. Pretty much, by now you should have guessed almost every chemical we talk about in the cell is going to be a protein. And proteins attach to antigens. And antigens are also usually proteins. So an example of an antigen would be this spike protein. Actually, just a little part of it is the crucial part. So the antigen is on the virus or the bacteria or the fungus or the parasitic worm. The antibodies are sometimes compared to guided missiles. You might remember that there are some antibody treatments for SARS-CoV-2. Now, your cells, when they make antibodies, again, antibodies are very specific. They're proteins, so we know that means they're going to work according to their three-dimensional shape. So an antibody that attacks the virus through the spike protein won't work on the virus that causes the flu or a virus that causes meningitis. It only works on this one protein. When this antigen attaches, think of it like a harpoon maybe on a whale. When this antibody attaches to the antigen, it changes its shape. And now the virus can't get into a cell. And if the virus can't get into a cell, it's kind of a sitting duck. Your immune system is going to be able to eliminate it pretty easily. Now, we also have some cells called T cells. B cells mature in bone marrow. T cells mature in the thymus gland. And they can destroy infected cells that can't be saved. And notice when I say infected cells, if we're talking about you having an infection, that means they're destroying your cells, human cells. Why? Because some cells can't be salvaged. So rather than lose a whole bunch of cells, the idea of these T cells is eliminate these ones that are already gone, that are beyond saving. T cells basically manage the immune response. There are some T cells that step up the immune response. These are called helper T cells. There are some T cells that basically monitor when you're fighting an infection and when you've completely eliminated the infection. There are some T cells that basically stop your response so that it doesn't spin out of control and go haywire. Another viral disease, HIV, is deadly because it affects helper T cells. And these are the main cells that kind of recruit all the other cells. So if you stop these helper T cells before they can actually recruit other immune system cells, it means your immune system doesn't work very well. And that's the basic problem in AIDS when you have an HIV infection. The immune system doesn't work as well as it should. Now, once you have an infection, some of your T cells and B cells make something called memory cells. And memory cells stay in your body and they still have the ability to recognize this antigen, this protein. And they still have the ability to make antibodies against this antigen, so they can respond. So the second time you get an infection, usually it's milder symptoms than the first. It doesn't last as long. Why? Because of all these memory cells, you're not starting from scratch like you were the first time. So the first time you get an infection, you don't know how to recognize this antigen. You don't know how to fight it. Once you learn how, your immune response is usually faster. It's usually stronger. It usually lasts longer. So it's a more complete response. So some people who have weak immune systems are more likely to get diseases multiple times, or vaccines don't really work with them. Now, because you have these memory cells, once you've had something, you have antibodies circulating in your blood. So you probably heard with COVID-19, there are some tests called antibody tests. With an antibody test, we can sample your blood. It's usually very quick, and we can find out if you've been exposed to the virus. Because what we can do is we take a little blood sample from you, we mix it with some of SARS-CoV-2. We put some of the virus in there. If you have the antibody in your blood, then the antibody and the virus will kind of clump together. Kind of looks like a scab, except it's on a smaller scale. If you've never had the virus, we don't see this clumping response. So we can see if you've been exposed to the virus. We can also literally count how many antibodies you have in a certain volume, so we can see what your level of antibody protection is. So there are 4 basic types of immunity. Natural active immunity. This is what happens when you get infected with a pathogen. A pathogen is something that causes a disease. So if you get infected with a pathogen, for instance, if you get a cold, colds are caused by viruses. If you get a cold, you'll make memory cells to respond to that virus so that the next time you get it, you probably won't even get a cold from it. You'll make antibodies, and this will be what helps you not get it the second time. So you might think, okay, I've had lots of colds during my life. How come if you make memory cells and you make antibodies against it, how come you keep getting colds? Well, the bad news is there are lots and lots of viruses that cause colds. I read one source that says there's over 200. I'm not sure how accurate that is because I've seen different estimates. But, you know, if you think about what that means, if there's exactly 200 viruses that cause colds, That means that after you get 200 colds, that could take a while, but after you get 200 colds, you should be immune to colds. You should never have to worry about getting one again. So this is natural immunity because you get this naturally. You actually catch a disease. It's active because you make memory cells and antibodies. There's also artificial active immunity, which is sometimes called vaccine-induced immunity. And this is what happens if you get a vaccine. And a vaccine sometimes is part of a virus or part of a bacteria cell, or it could be dead cells or dead viruses. And it's something that stimulates your immune system to make antibodies and also memory cells. Now, the idea of this, of course, is that with a vaccine, you don't actually get the disease. So, for instance, if you get a vaccine against COVID-19, then instead of getting the disease the first time, and then you have immunity, the first time you're exposed to it, you don't get the disease because of the fact that you have already had a vaccine. Now, if people have a very weak immune system, their immune system might not learn to recognize the virus when they get a vaccine. So the vaccine might not really work for them very well. Now there's also, now notice this is artificial because, vaccinations aren't something that we see in nature. There's also something called natural passive immunity. And this is what happens when a mother passes antibodies to the fetus across the placenta, or when a mother passes antibodies to the baby during breastfeeding. This can protect the fetus or the baby, but the problem is, what antibodies get passed to the fetus or the baby? Well, that depends on what antibodies the mother has in her blood. So the fetus or the baby is never exposed to the disease. So their immune system doesn't make antibodies or memory cells. So this protects them, but for a fetus, it only protects the fetus while it's attached to the placenta. For a baby, it only protects the baby while it's breastfeeding. Some of the memory cells that you make can last decades. It depends. Different illnesses stimulate memory cells. Sometimes the memory cells are stronger, sometimes they're weaker. Sometimes they only last a few years. And then finally, there's something called artificial passive immunity, which we don't see very often. But remember, we said natural passive immunity, you get antibodies from your mother. You don't learn to make your own, you don't make your memory cells. Artificial passive immunity is similar, except you don't have to be a fetus or a baby. We give you an injection of antibodies. And typically we do this because a disease is fairly serious. It's serious enough we don't want to wait because you might not survive the first time you get the disease. For instance, there are some snakes whose bite is so poisonous that the chances that you'll die are very good. So what do we do if you get bitten by these snakes? We give you some antibodies. Now, how do we get antibodies? Because you get the way that your body starts making antibodies is you get exposed to the disease. Well, we could get them from a person who got bitten by this snake or spider or whatever the poisonous animal is and survived. They've got antibodies, but there's another place we can get them. Sometimes other animals aren't poisoned by the same things that we are. So if they're not poisoned by the same things we are, that means that we can basically keep them in a lab, infect them with a certain type of poison, let them make antibodies, and then, kind of like we talked about a moment ago, we can take a blood sample from them and we can purify those antibodies. So this way, if you get bitten by a snake, like a cobra, where that can be fatal, instead of waiting to see if you survive or not, we could potentially give you antibodies and then you survive. Now, the bad news is, if you got bitten by a cobra, you didn't get the antibodies, but you survived. Now you would have memory cells, you would make your own antibodies. If you get bitten by a cobra today and we give you a shot of antibodies, you survive. But the next time you get bitten by a cobra, since you didn't actually have an immune response, we gave you those antibodies, you're going to have to get antibodies again or take the risk of, do you survive, do you not survive? So viruses are very common. Keep in mind what I said. They can infect almost any living thing. Viroids are very different. They're small, single-stranded molecules of RNA, that are circular. Now, we don't hear too much about viroids, and this is because all the viroids that have been discovered up to this point infect plants. So viroids get into a plant cell, they attach to the DNA in the nucleus, and then they interfere with that plant cell making proteins. So one of the most important viroids, just because they don't infect people, doesn't mean they aren't important. One of the most important ones is a virus that causes potato spindle tuber disease. It's actually called the potato spindle tuber disease virus. But what this virus does is when it infects a potato plant or a tomato plant, potatoes and tomatoes are actually closely related. When it infects one of these plants, the plant grows kind of tall and thin, so it tends to fall over. And sometimes the fruit also is elongated. And it's actually estimated that around the world that this probably causes, hundreds of millions of dollars worth of economic damage to potatoes and tomatoes every year, where the crops are ruined. As I say, we don't hear too much about viroids because they infect plants. Prions are actually just proteins. They're proteins that have the correct primary structure. So they have the right amino acids in the right order, but something goes wrong and they fold into an incorrect three-dimensional shape. And when they get into a cell, they can cause normally folded molecules of the same protein to misfold. So kind of like a chain reaction. And these improperly folded proteins form a clump called an amyloid. And the amyloid forms in the brain. And amyloids cause cell death. So in other words, this kills cells in your brain. Now, the only prion infection that most of us have heard of is something called bovine spongiform encephalopathy, or BSE, which is what we commonly call mad cow disease. Now, mad cow disease is found mostly in cows, as you might guess, but occasionally, very, 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 very rarely, it's passed from cows to people. Notice that your odds of getting hit by lightning are better than your odds of getting mad cow disease, much better actually. And prions are also suspected to cause something called Creutzfeldt-Jakob disease, which is actually a genetic disease, or something called fatal familial insomnia. So this is, again, a genetic disease. It runs in families, as you can tell from the name. And in this case, a person has so much trouble sleeping, it's eventually fatal. And another disease called Kuru. So these are all believed to be caused by prions. There are no treatments or cures for diseases caused by prions. So if you get a disease caused by a prion, they seem to be fatal. Now, diseases caused by prions, even though they're fatal when you get them, they're very, very, very rare. So they kill maybe 5 people in the world a year, not very many. The reason we're interested, though, is because there are some diseases like Alzheimer's disease, Parkinson's disease, ALS, amyotrophic lateral sclerosis, that are also caused by amyloids. Remember, this clump of proteins up here was an amyloid. Now, I want to emphasize right now, there's no evidence that Alzheimer's disease or Parkinson's disease or ALS is caused by a prion. But because they are caused by a similar clump of proteins, we'd like to learn more about how prions work, because that might give us some clues about treating these diseases. Right now, we don't know too much about them. Are there any questions about viruses or viroids or prions? Okay, if not, we will stop sharing screen. And if you have any questions, we'll see if we can answer those before you go. Otherwise, I will let you go for now. Okay, if there's no questions,